pop it in the chat box. Thank you. Feel free to pop it in the chat box, or you can reserve your question until the end of the session. We can have the face-to-face conversation after this. Okay. I'm hoping that I'm sharing the right screen, so Shafina can let me know. Try to make it full screen. Am I sharing the right screen, the full screen of the slide, Shafina?
Yes, correctly.
Okay. All right. So let me arrange my window. I have 3 screens in front of me, so I can see everything. Okay. Check that. All right. So yes, Assalamualaikum and good morning again, everybody. So thank you so much for joining us this morning. I'm excited to share what is all about enzymes and genetic engineering, because my background is actually biochemistry and molecular biology. So I have never worked with enzyme engineering personally, but all the tools that I'm going to be sharing with you are the tools that I use every day since I started my research career. I think back during my master's time, that was when I first learned about enzymes, and I've been using different tools until this date in order to improve our understanding of the microbiology and molecular biology of the world around us. Okay, so the title that I'm going to share with you today is on harnessing the genetic and genomic innovations for industrial enzyme technology. And as Sharfina has introduced to you before, I'm currently affiliated with the Department of Biosciences, Faculty of Science, UTM. So we first met during Sharfina's MSc time at UTHM. Just a little bit of history. She contacted me to inquire about assisting her with SDS page, which is a protein characterization method. So we have the system in our lab. So I said, no problem, come to my lab and see how we can help you. And I'm happy that we are still collaborating to this day. Hopefully the collaboration will continue for longer. Okay, so before we talk about the topic itself, let me introduce to you UTM in case this is your first time hearing about our university. So we are actually a leading research university in Malaysia. So in Malaysia, we have 5 research universities. UTM is one of them. Our focus is on engineering, science and technology, and currently we have about 1,500 academic staff and 92% of them with a PhD. And the history of UTM, we started way back in 1904 as a technical school, focusing on technical and engineering courses. And then we were upgraded to university status, first known as Institute Teknologi Kebangsaan, or ITK, in 1972. And we became UTM, University of Technology Malaysia, in 1975. And in between here is when we moved our campus from Kuala Lumpur. We still have the Kuala Lumpur campus. We call it a city campus. So we moved to Johor Bahru as our larger main campus. So we have been in Johor Bahru ever since. And we got the research university status in 2010. And these are all the different strategic plans that UTM have put in place to direct our strategic planning. And we are at the end of the Envision 2025 strategic plan. So we are now busy drafting a new plan to guide us all the way until 2030. So some pictures of UTM. So I would like to take this opportunity to invite everybody to come to UTM. Maybe Sharfina can help to arrange an academic visit for the students to come here and visit us. Or if you would like to come here for academic exchange for 1 or 2 semesters also, we can try to arrange that. So here is our main gate at Johor Bahru campus, and you can see the we call it the magic Quranic verse that we have here. Dan dikatakan Allah, Ya Allah, tambahkan ilmuku. So this is like the mantra that we use every day to guide our mission in educating the community around us. And this is the main mosque at UTM. I would say one of the most beautiful university mosques in Malaysia. So this is the KL campus. And we also have a third campus in Pago, near where Sharfina did her master's, focusing more on agricultural technology. And some more information about UTM in numbers. We have about 24,000 undergraduate, 24,000 students. 16,000 of them are undergraduates, 3,000 masters, about 4,000 PhDs, and we have about 4,700 international students. And for staff, we have 4,800 around that. And as you have seen before, the academic staff, we also have professional and supporting staff, about 3,000 of them to assist us in administration, laboratory, and all the supporting supporting centers. And so far, we have more than 200,000 alumni in Malaysia and all over the world, coming from 87 countries, and I think it's increasing day by day. So the Faculty of Science is actually one of the faculties in UTM. You can see here we are very heavy on engineering, but the Faculty of Science is one of the non-engineering technical faculties. The others are computing. So we also have the new faculty, artificial intelligence, and built environment surveying. So these are the non-technical, the social sciences and humanities and management. In Kuala Lumpur, we have the Malaysia-Japan International Institute of Technology and the Business School. So this is the newest new faculty, which split from the Social Sciences and Humanities, which is the Faculty of Educational Sciences and Technology. It is basically the education school, so training teachers. So for the Faculty of Science, we have 4 departments, the Biosciences, which I belong to. And you can see here, these are just the panels, the loosely affiliated panels that we have. So our research can be affiliated to one of these or slightly different from this. So these are just the academic panel, we call it. And then we have the Department of Chemistry, Mathematical Sciences, and Physics as well. So this is our organizational structure. Our dean, Associate Professor Dr. Shafinas, and then we have 2 deputy deans for academic and student affairs and research, innovation and development. And this is where I am right now as the manager for external and global engagement. So we have the manager for quality and strategy as well. And these are the lineup of the head of departments for the 4 departments. So you can see there are a few other non-academic staff assisting with administration. For example, Madam Safriza here. And then we have Aunty Sharil, Chemist Puan Zubaida, Dr. Azwani, and the others assisting with the administration. So I think we can skip this one, maybe because Sharfina has kindly introduced me just now, quite detailed. But I just want to highlight my other interests here. So other than research in the lab, so I'm interested in chemical safety and security education. I also got involved in various science communication and advocacy activities. We do a lot of STEM education and outreach programs, and also science diplomacy, which is policy making at international levels on scientific aspects. So we call it science diplomacy. All right. So let's do a little bit of fun quiz. So it's a rapid fire round. Don't take too long thinking about it. I just want to see your understanding on the topics and try to wake you up if possible. So please give me the answer through the chat box. we have A, B, C, D options, A, B, C, D, some A, B, C only. So just fire away your answer in the chat box. Don't take too long. Don't think about it. I won't penalize you if you got the answer wrong. Just want to see whether you'll be able to get the answer correct. Okay, shall we begin? First question. All right, what is the definition of genetic engineering? A, B, or C? Okay, we have two answers, 3, 4. Keep it coming. Sharfina is not included. You are not allowed to answer. Okay, we have 10, 9 or 10 answers. We have 69 students here, you remember? So I hope to see 69 answers in the chat box. Okay, so this is very easy. Everybody says B. So genetic engineering is making changes in the DNA code of living organisms. All right, let's go to the next question. What made genetic engineering possible? The Dolly? Is it Dolly? Is it Human Genome Project or the DNA sequencing? Maybe I should have a prize of who got the first answer the fastest. But yeah, you are all the way in Jakarta, so it's a bit difficult to send the prize. Or not Jakarta, in Jogja. Maybe the next time I go to Jogja, I can bring something to the winner. Okay, so many people say C, DNA sequencing. Yep, that's one of the important discovery that made genetic engineering possible. Okay, anyone else say it differently? Anyone wants to say Dolly? Nope. All right. So Dolly is another important invention to advance genetic engineering, but that is not the pivotal or the important technique that makes genetic engineering possible, right? All right. So next question. What was the first organism to be successfully modified via genetic engineering? So try to answer without having to Google. A, plant, virus, bacteria, or mouse? So this is a bit tricky, looking at the slowness in the answers coming in. So Yuda answered B, virus. Anyone wants to say something else? C, bacteria. Anyone wants to say plant or mouse? No idea. All right. So the answer is actually bacteria. C. Yeah. So Zul, Kifli, congratulations. Do you know the first bacteria to be genetically modified? It's our the most famous bacteria of all, which is E. coli. As we know, it's a common bacterium. And the breakthrough actually happened in 1973, if I'm not mistaken, by Boyer and Cohen. So that was the first organism to be genetically modified using recombinant DNA technology, which includes insertion from one organism, non-bacteria, or maybe a different bacteria into E. coli. So that discovery actually laid the foundation for modern biotechnology, as including GMOs, we have gene therapy and production of synthetic insulin and many others. So the answer is C, bacteria, the first organism to be modified via genetic engineering. All right, so the next question, we have a few more. Keep the answers coming. So the majority of genetically modified crops are grown in which part of the world? Is it the Americas, including North and South? Or is it Europe, Southeast Asia? Do we have GMOs in Southeast Asia? Or is it China? So tell me the answer, A, B, C, D. So we have some A's, we have D. Oh, it's gone. Just now I saw D as an answer, China. Don't worry, don't feel shy to give me a wrong answer. We're just having fun with scientific questions this morning. Don't be too pressured to give the correct answer. Okay, so yes, the answer is the Americas, where most genetically modified crops are grown, which is the North and South America. I think they account for 85 to 90% of the world's GM crop in terms of the area. So we do in Asia have genetically modified crops, particularly in India and China, and also Africa, but much lesser extent compared to the Americas. So in Europe, because of the stricter regulation, in Indonesia or in Malaysia, we have GM crops being cultivated. All right, next question for you. What is the primary reason for genetically engineering microbes to produce enzymes in the industry? Now we are starting to talk about enzyme. A, B, C, D, another easy question. Do we want to make them glow in the dark or make them taste better? Well, we do eat microorganisms, right, in our yogurt, but do we genetically engineer them? Or to produce large quantities of useful enzyme? This is another easy giveaway question for you. Mostly used for producing large quantities of useful enzymes efficiently. We don't use microbes as pets. Not as far as I know. Maybe some people use them as pets, but not me and not Sharfina. Not yet, maybe. Okay, thank you guys. So the answer is C, to make large quantities of useful enzymes. Next question, just a few more. So which genetically engineered organism is widely used to produce rennet? You know rennet, the enzyme used for cheese making to make the milk go curdled. So we use rennet to make cheese. So which GMO, which GM organism is used to produce the enzyme? Is it yeast, E. coli, bacillus, or cow? What's the answer? A, B, C, D? So we have C, everybody saying bacillus. No one wants to say cow. Cow makes milk, right? But not producing the enzyme. So what's the answer? B. Oh, B and C. Unfortunately, the answer is A, yeast, because one component of rennet is the enzyme chymosin, and a yeast, the Saccharomyces cerevisiae, is used to produce chymosin, which is one enzyme in the rennet enzyme mix. Okay, so is it the final question? No, the second last question for you. Which enzyme produced through genetic engineering is key to stone washing denim jeans? Is it lipase, cellulase, amylase, or DNA polymerase? What's the answer? So Yuda said B. I hope that's the answer for this question. Yeah, Yuda. B. Anyone else wants to say different enzyme? Lipase, maybe, or amylase, or DNA polymerase? Mostly B. Yeah, you are correct. Well done. It's cellulase. So we know why cellulase, because of the cellulose component in the genes. So this enzyme is used to create the stone washing effects on the denim genes. Okay, so last question for you this morning. So in detergent formulation, which genetically engineered enzyme helps break down protein stains like blood? I think this is another giveaway question for you. Very easy. What's the answer? You have protease, lactase, urease, or synthase. Yeah. So protein, of course, the enzyme working on protein is protease. The protease sometimes added to biological detergent to break down biological components in um, soil or stain fabrics like blood. However, nowadays, I think biological detergents are being it's not replaced. You have alternatives whether to use biological detergents or non-biological. I'm not sure whether in Southeast Asia we bother to check whether it's biological or non-biological. But in the UK, they are very particular because some people are allergic to enzymes in biological detergents. So the manufacturer must clearly label their detergents or washing powder biological or non-biological. So if people are sensitive to biological component in the biological detergent, so they have to choose the non-biological ones. I don't think we care that much in Malaysia or Indonesia because probably we don't have the same sensitivity to the enzymes in the detergents. Okay, so thank you very much, everyone, for participating. So I can see a few people actively participating. Hopefully, the others are also listening to me talking this morning. Yeah. All right. So the lecture outline. Firstly, I would like to introduce to you about gene and genetic engineering. So maybe some of the topics that I'm going to be introducing is you have heard of it before. It's very basic. Maybe you have learned about it in your different courses, but I think it will be good to have to start from the beginning to give you some context to refresh your memory. And then we go to a more related and more advanced topics about enzyme engineering. So I will then, after introduction, I will then introduce to you the techniques. Some are simple techniques, some are more up-to-date techniques on enzyme engineering. And then we'll be looking at a few case studies, some interesting papers that I could pull from the literature to share with you, the scientific advancements that lead to the important moments in enzyme engineering. And some have even been commercialized widely. And then I'll give you the summary of today's lecture and then some future outlook of what I think the field is going to go in the future. Okay, so what is genetic engineering? I think everybody knows this. It is actually a set of technologies used to change the genetic makeup of cells and to move genes across species boundaries to produce novel organisms. So a simple modification in the genetic makeup of any organism will give you a novel organism. It may not look like it is different, but as long as the genetic makeup is changed, you can say we are making a new organism. So you can call it gene manipulation, gene cloning, which is a very classical term now already becoming classical, or recombinant DNA technology or gene modification. So of course, it is based on genetic information, as we know, encoded by DNA and arranged in the form of genes. So genetic engineering or GE is a very valuable resource. that can be used in many ways to achieve our goals in many different fields of science and medicine. Okay, so we know this also. What are genes? Genes are made up of DNA, so we call the genetic makeup as the genotype of the organism, and the genotype will give rise to the trait, what we can see. So we call that a phenotype. And high school science or high school biology, they consist of pyrimidine bases, the TNA, the TNC, and the purine bases, the A and G. And so genes are the component of the DNA that we normally move to another organism that will also transfer the phenotype as well. So a little bit of history of genetics and gene manipulation. So in the 1900s, so that was when Gregor Mendel was investigating genetics by observing the color of the pea flower. So that's how he discovered those traits are passed to the offspring. And then starting on from then onwards, the field expanded very rapidly. And in the 1940s to the 1950s, that was when these two guys, Watson and Crick, discovered the structure of DNA using the X-ray data by Rosalind Franklin. And I think that was when we have explosion of discoveries, when people discovered the structure of DNA. It just goes on exponentially from there. So we also have gene manipulation era where many techniques were developed starting from the 1960s to the 70s with many different applications. And then the human genome was sequenced during these times, and the post-genomic era, and also the discovery of epigenetics, which is the study of heritable changes in gene expression that is not caused by the DNA sequence, but more on other components that is affiliated or associated with the DNA molecule. And now the most interesting era in this field, I would say, is the omics era. We have the different omics tools like metagenomics, metatranscriptomics, or metabolomics that can guide genetic manipulation in the field of protein and enzyme engineering. So this Professor Francis Arnold was one of the important innovator in that field. And of course, the in thing currently is a CRISPR-based gene editing discovered by Jennifer Doudna and her colleagues. So we don't know where this is heading, but looking at the interesting tools that have been developed so far, we can say that there are many things that we can do with the tools that we have, and the opportunities, I would say, basically endless. Okay, so everybody has heard of gene cloning, and just to refresh your memory, this is based on myself. What are the 4 steps in gene cloning experiment? So I divide it into 4. Firstly is the generation of DNA fragments, and after that, we can join the DNA fragments that we generate to a vector or a carrier molecule, and then the carrier molecule with the gene can be introduced into a host cell for amplification. And finally, after we introduce it into the host cell, we need to know how to select whether the sequence is actually present in the host cell or not. So let's go through this one by one. The generation of the DNA fragments. So you can get any gene from any organisms that you want, from microorganisms, from animal cells or plant cells. Get a gene of interest, and normally what we do, as you know, is to digest it with restriction enzymes. So you digest the gene and digest the vector. In bacterial case, I do a lot of bacteria based studies. So in my case, what vector that I use is often plasmid. So we can ligate the gene to the vector, in the first stage, and then we get a recombinant DNA, the vector containing the gene. So this is if your ligation is successful. So this is just a simple example of a plasmid containing a region that we call multiple cloning site, that you can insert any gene that you want, and a few marker genes like ampicillin resistance to see whether the transformation is successful. And of course, all plasmid must have the replication origin. So in the case of gene cloning, it is often shown as a circular molecule like this, because as we know, plasmid is circular, just like the chromosome. And in bacterial cell, we know that there is a plasmid that exists independently of the chromosome. So the easy cloning experiment would be to insert the gene of interest into the plasmid and transform it into the bacterial cell instead of inserting into the chromosome. But there is a way to insert it into the chromosome if you want. But the easiest, the first studies done is typically using plasmid. So once you have joined your gene of interest to the vector or carrier molecule, then you can introduce into the host cell for amplification. So in my case, I use E. coli a lot as a host cell. So then we can do what we call transformation, transforming the recombinant molecule into the E. coli cell. And there are many ways to introduce the recombinant molecule into E. coli cell. Commonly used one would be transformation. So transformation is very easy to do using a chemically competent cell, or you can also use electric shock to do the transformation. If you're talking about plant cell, maybe the more widely used method for transformation is projectile gun, or for mammalian cell, maybe micro injection would be widely used. And for virus as well, for bacteria transformation as well, we can use a virus to deliver the foreign DNA molecule. And now we have the transgene, or the foreign gene inside the host cell. So this is just an illustration. Normally it is in a circular plasmid. So after we have done the transformation, there must be a way of knowing whether our recombinant DNA has been successfully transformed and is stable and amplified inside the host cell. So we have to have ways of selecting the required sequence. In the case of bacteria, again, there are three widely used methods to see whether the transformation and the recombination is successful. Firstly is to see the antibiotic resistance. So it's very simple. Just grow your bacterial cells on a Petri plate with antibiotics. So the cells that does not have antibiotics could not grow. And we can also use blue-white screening, which is based on the disruption of a gene. So if the gene is disrupted with the insertion of the foreign gene fragment, the colony becomes white. If it's not disrupted, it becomes blue. So you can see the blue ones are the ones that does not have our gene inserted, just the plasmid vector. Or we can even do colony PCR, because sometimes we can have false positive. The white ones can also be a molecule transformed without insertion. So doing a colony PCR to see which of this colony contains our gene of interest is also possible. So colony PCR is just picking up any colonies that we have here and then extract the DNA straight away, and then we can send for sequencing to see whether our gene is present inside the plasmid in the host bacteria. So yes, if you are using different hosts like plant cells or mammalian cells, there will be different methods of knowing whether your transformation and combination is successful. So let's dive into some genetic and genomic techniques. There are so many genetic and genomic techniques available. right now. So I just pick from the most basic to the most commonly used ones in especially industrial enzyme applications. So of course, I think everybody knows about gel electrophoresis. You have probably played with gel electrophoresis in the laboratory, and it is one of the classical tools in genetic engineering. It is based on the migration of charged molecules, which is the DNA molecule, through a matrix, which is the agarose gel or polyacrylamide gel, in an electric field. So the movement is based on the movement of electricity and ions. And the distance travel in the matrix depends on the molecular weight of the DNA. So we can use either agarose or polyacrylamide to observe the DNA fragments in the mixture. So the big ones will be on the top, the small ones will be at the bottom. So normally for my students, I will dive deeper into agarose gel electrophoresis, including what buffer that we use and how to determine the size of the DNA fragments that we have. But for this purpose, this morning, I don't think we have time to go that deep. So just to refresh your memory about gel electrophoresis. And then we have the most famous, probably, molecular tools, polymerase chain reaction, or PCR, which allows amplification of a minute amount of specific DNA sequence. So from one molecule, we can actually create millions of molecules, same one, in just a few hours, so that it is easier to be detected, to be studied, and to be used. And this method, for your information, was discovered back in 1983, which is the year I was born. So PCR is close to my heart because it is as old as me, which is 42 years old. So I'm giving you my age right now. So we know that PCR has three important cycles, which is denaturation, annealing, and extension. And then it goes back to denaturation, annealing, and extension. We do that 30 cycles, and we can get up to 1 billion copies of the DNA or of the gene fragment that we want to amplify. So, of course, the PCR reaction requires thermostable DNA polymerase because we use up to 94 degrees or 95 degrees for denaturation, and we don't want to keep adding enzyme after every cycle. So we use a thermostable DNA polymerase to do the extension of the DNA molecule. And originally, the DNA polymerase was isolated from a thermophilic or organism that lives in a hot environment, which is a hot spring, Thermus aquaticus. This is the bacteria where the first polymerase was isolated. So we call it Taq polymerase. But nowadays, there are many advanced types of polymerases that have been engineered depending on the application. So we have polymerases for amplification. We have polymerases for DNA sequencing. We also have polymerases for modified oligonucleotide synthesis. So each of these polymerases have been specifically engineered to be suitable for the different applications. So this involves the molecular tools that we are going to be discussing this morning. All right, DNA sequencing, your answer just now, the most important invention that gives rise to genetic engineering. So there are a few generations of DNA sequencing up to now. We have first generation, we have second generation, and nowadays the third generation DNA sequencing are more widely used as well. So the first generation are the reaction that is based on the NTP addition and then visualization of the reaction using electrophoresis. So the most famous would be the Sanger sequencing. I still remember during my master's time where we sent for sequencing of a single gene, and then we got back results of electrophoretogram that different colored peaks to see what is the identity of the nucleotide at that particular site. So normally for first generation sequencing, we sequence a single gene in a single organism. And the fragments generated is about 500 to 1,000 base pair. So that's why we need to have two primers to do the first generation sequencing. So in the example of using it for bacteria identification, we target the 16S gene fragment, which is about 1,500 base pair. And we have the forward primer will give about 1,000 fragment, and the reverse primer will give about 1,000 base pair fragment in a different direction. So that will be able to cover 1.5 base pair gene length. But nowadays, it has evolved into second generation sequencing because we don't do Sanger sequencing that much anymore. So we are using more advanced DNA sequencing technologies, and the most commonly used one here is Illumina. So Illumina allows us to do a sequencing reaction in parallel, where you can do many runs at the same time, but the fragments generated is much shorter. So the second generation sequencing is normally used for metagenomics, where you want to sequence many DNA molecules in the same in the same mix. So nowadays we don't really isolate bacteria anymore. If we want to study that environment, what we do is just to extract all the DNA molecule present in that environment. We call it metagenome, and then we sequence all at once, so we can know all the single members present in that environment. So it gives the short fragments that we have to use computational tools to align and to analyze. But nowadays, the more recent sequencer is able to generate long fragments. So it's no longer 1,000 base pair fragments or 500 base pair fragments. It can be tens of kilobases length fragments. So normally what we do is to combine long read sequencing with the short read sequencing that will give us a very in-depth and detailed information about the microbial community in that particular environment based on the DNA sequences from the total DNA that we extract. So normally we use Oxford Nanopore. And Oxford Nanopore, another advantage of this is that it's very small. There is a type of Minayan sequencer, which is very small, and we can bring it to the field. So you can go to the most remote area and be able to do DNA sequencing. So it's very interesting. All right. So another method is to do mutagenesis, which is the process of inducing mutations or changes in the DNA sequence of an organism. Among the different types of mutagenesis include random mutagenesis, site-directed mutagenesis, site saturation mutagenesis, and combinatorial mutagenesis. So this is another method that is close to my heart because I did mutagenesis almost every week during my PhD, in particular the site-directed mutagenesis. So what are they? Random mutagenesis are mutagenesis introduced through targeting without targeting specific residue. So how do we do that? Using error-prone PCR. So error-prone PCR will use a polymerase enzyme that is not very accurate. So it always introduces errors. So you may wonder, why do you want to have error in our PCR? Because when we have this random situation, sometimes when we do PCR, it will give rise to mutation at certain sites. And this mutation actually gives the advantageous property to the gene and to the enzyme generated. So that's why random mutagenesis is quite useful. Sorry. So we can use error-prone PCR, or we can use UV or chemical mutagens to do the random mutagenesis, or using a transposon, which is a jumping gene to just jump at random locations in the DNA molecule and create this mutation. And site-directed mutagenesis, as the name implies, we target a specific site in the DNA sequence to change the DNA sequence. So what method can be used for this purpose is PCR based again. So this is what I used before, using quick change kit by now it belongs to Agilent company. You can also use oligonucleotide directed mutagenesis. This is non-PCR method, or more recent, you can use CRISPR-Cas9 tool to introduce site-directed mutation. Or you can do site saturation mutagenesis, where you change that particular amino acid in that specific position to all 20 amino acids present to see which amino acid is the best for the enzyme activity, stability, and so on. And combinatorial mutagenesis is a combination of the different mutagenesis available. All right, so I'll introduce you to site-directed mutagenesis a little bit more, since I've done this many times before. As we have seen before, it is to introduce mutation at a site-specific location in the DNA molecule or in the gene that we are interested in. So how do we change, or how do we plan for the mutation? It's based on the codon information. So we know that the codons are the 3-letter codes that translates into the different amino acids. So you can change from one amino acid to the other by changing 1 or 2 letters in the codon code. It is normally used for proteins and enzymes, whether to improve stability or change the specificity of the reaction of the enzyme, or reduce toxicity when we do overexpression in the cell. So one example of a commercial kit that we can use is called Quick Change Kit. This is what I used before. Now it is owned by Agilent Teknologi, a large chemical and instrument company. So how does it work, this Quick Change Kit? It is based on PCR. First, what we do is to have 2 primers, forward and reverse here, which contain